Testing. Okay, I have to make a part two. I just noticed that it cut me off at about 15 minutes, so I've got about 20 minutes that I need to add into here. So I'm trying to make up for the time that I missed it, or the time that I got cut off. Um, and I don't remember, because I didn't hear the end of the video clip on the other one, but one of the things, the slide right before this, I had said that the very first genes identified were in a nematode, and almost 40% of the genes identified in the human genome are the same as the bacteria feeding Caenorhabditis elegans. So that was the very first. A nematode was the very first genome identified, and it's been used for biological research since then. One of the main things you're going to look for with plant materials are wilting, stunting, and yellowing, mainly because of damage to the root system. So you're going to look for hot spots. You might find circular regions in the field. In order to prove that it's nematodes, you need to look at the soil, the roots, and underground parts to determine what's going on. This particular slide was taken from page 72, and it shows the morphology, and relative size of some of the important pathogenic nematodes. Longidorus is the longest, slimmest one, and you can see it's a plant-feeding one because it's got a long stylet here, its throat, and there's its long intestine, and it's a very, obviously for its name, it's a long, slender nematode. Ephenolchoides is another one of the plant-damaging nematodes, and we'll talk a little bit more about that. Praedolenchus are our lesion nematodes. Meloidogyne are the root knot nematodes, and Heterodera are the cyst nematodes. And the main thing is you want to look and see whether they are ectoparasites, where they feed from outside the plant and they move migratory feeding from plant to plant, or they're sedentary remaining on the same plant, or they're endoparasites, where they feed from inside the plant, where they go up and migratory, where they move within and feed on the tissues of the same plant, or they stay within the plant and stay at the same feeding site. So probably our biggest and most damaging nematode is going to be the root knot nematode here. And I think the next picture, this actually shows the cross-section of a giant cell system initiated by the female root knot nematode. And basically, the root knot nematodes are sedentary endoparasites, and these are the second stage juveniles that are penetrating just behind the root cap. And this permanent feeding site is called a giant cell, and it's within a gall. So that's where she actually creates the gall, and it's formed in response to the secretions that are given off by the nematode. And the nucleus continues to divide, but the cell division doesn't occur, so it just gets bigger and bigger, and the cells swell. So basically, some of the juveniles are going to grow and molt, and they're going to become round adult females. And what they say is they lose their vermiform, which means they're worm-shaped, because they become a gall instead. And root knot nematodes have the widest host range of about anything. They're non-host specific, or excuse me, no, that's what I meant to say. They're non-host specific. Root problems are easily visible on root crops like carrots and beets and potatoes, and but worldwide, probably the root knot nematodes cause more crop losses than any other genus of nematode, especially in vegetables and flower crops. They're probably one of the biggest ones. I don't have a picture of the cyst nematode on the slides, but there is one in the text on page 73. And it just looks like a big, like a peach, and you can see all the nematodes inside, basically being released from the cyst nematode. And this would be another sedentary endoparasite. the difference between root knot nematodes, which are not host specific, and cyst nematodes is cyst nematodes are host specific. So you're going to have the brown cyst, or I should say the soybean cyst nematode specifically feeds on soybeans. And the most important crops where we have problems with cyst nematodes are some of our cereals, soybeans, sugar beets, potatoes, and tobacco can be a problem too. Okay, so moving on to this slide, this is a picture of a lesion nematode, which is a Praedolenchus species. And these are migratory endoparasites, so they actually move within the plant. So they're known for the lesions that they leave after they've gone through the plant. So they feed on the plant, they're moving through, and then you see the dark spots that die after they go through. So the dying cells then, in a lot of cases, are invaded by fungi, so that's why they get dark. And they have a wide host range. And where the arrow is on this slide, this is the stylet of the Praedolenchus nematode, which is pretty cool. Okay, there are some other burrowing nematodes similar to the nematodes, similar to the lesion nematodes, the Rhodophyllus species, and they're more migratory endoparasites. But these are more in tropical crops like bananas and citrus. So we don't see as much damage from those. Okay, so moving from those parasites, we're going to go to the migratory ectoparasite nematodes. And here we've got common names like the dagger, which is the Xiphonema, sting, Benolamus, and these actually refer to the shape of their stylets. So dagger or sting. Stubby root refers to the symptoms that the nematodes induce. So the stubby root nematode is a Trichodorus. And we've got a wide range of hosts in the ectoparasites, and all life stages, all the juvenile stages and adults are all infective. And this is a picture of an ectoparasite ring nematode, Mesocriconema xenoplax. And the criconemas are very distinctive in these ridges that go all the way around them. You know, it could be like a small beehive, but that's all the way around them. And they're the only genus, the Mesocriconema, that have those. There are many different species within them, but it's going to go to the genus, and then you would look at the stylet and the rest of it to go to the species of them. On page 74 in your text, it actually separates out the common root-infecting parasites as to sedentary, semi-endoparasitic, and migratory. And then it also goes into the common root-infecting ectoparasites as well. And probably more than anything else, I'm interested in you knowing the common names. You can relate them to the Latin names later on so that you know what they are. Most commonly, you're going to see, a nematode report is going to have the Latin name on it as well, but they should give you the common name. Trying to think of where I want to go with this. We do have some nematodes that affect plants above ground. So my foliar nematodes are going to be Aphenolcoides, which was that middle one I showed you on that picture, and they're going to be migratory and move from the soil to the lower leaves of the plant. So this might be something like chrysanthemums, and basically the lesions are usually stop right at the major leaf veins because they can't go through the vein. It's a very coarse material, so they'll stop at the vein. A lot of times the foliar nematodes can invade through the stomata, but again, it goes through a lot of different plants. We can actually have some seed gall nematodes. The Anguina species are migratory. We've seen them in Annual bluegrass, but they can cause stem and seed galls on grasses. They usually do not infect dicots. It's mostly the monocots. But they're going to feed on the developing seeds, and they're going to cause seed galls that are filled with nematodes that instead of the seeds in the seed head. I'm trying to think of what else is important. One of the things that we need to look at, and I probably should go back to the other nematode. I'm getting a little bit ahead of myself. Nematodes don't have specialized survival structures. So if they don't have a host, they're going to disappear, either by dry soils or winter freezing. But perennial plants, both crop and weed seeds, actually support nematodes in the winter. So you'll see survival with some of these species with various perennial weeds they're attached to. So when you look at a lot of the survival of nematodes comes from infected propagated plant parts, so bulbs, corms, tubers, rootstocks. And then by putting those into the soil, then we're introducing the nematodes into the soil along with the plant material because it's already in the planting crop. So nematodes can survive as eggs in the soil, but they will dry out in the winter in dry soils, and the population will decline quite significantly over the winter months. The cyst nematodes, the shells on their cuticle of the egg within the dead female body is very tough, so they're going to last through the winter. Okay, so if we look at this slide, the pine sawyer beetle is the vector of the pine wood nematode, and that causes pine wilt disease. Okay, so there is actually the tree pathogen is transmitted by the insects. So the pine wood nematodes are carried in the trachea of the sawyer's beetle, which is the picture I'm showing here, and its respiratory system, and emerge when the beetle feeds on a new host. So then the nematodes colonize the new host and cause the pine wilt. And they can travel in wood chips or unseasoned lumber, and several weevil species are vectors of the red ring nematode, and they can actually lead to the red ring disease of coconut palms. So it's important to control these things, and one of the ways that they can do this is to make sure we don't have nematodes or insects being transformed or transmitted in wood, is to take them and kiln drying the wood so that kills off anything that might be living in that wood, because that can eradicate the pine wood nematodes before it gets exported to any countries that don't have that pathogen. So that's what APHIS is for, is to help with that. Okay, so the next one is how do I diagnose nematode diseases? The first thing we're going to want to do is look at any of the above ground symptoms, and so areas where they're not growing very well, where it could be wilting, where it looks like I have a water deficiency, and basically it could be caused by root feeding nematodes, and it could actually complicate which would allow in root infecting organisms, which would actually complicate the diagnostic process. But the nematode may have been the first faction that actually caused that. So nematodes, which infect above ground plant parts, so seed gall, stem and bulb nematodes, can be diagnosed by tearing apart some of the affected tissues while looking through a dissecting microscope. When we go into roots like this is a lesion nematode. Remember that it's got its name by, if the tissue is dark and dying, then the nematode has probably come in here and it's moving up this way. So this is already gone, so the active nematodes would actually be up in here, the area that's still white. And the nematodes have killed this area. We've got a disease coming in already, the cells are dying, and the active nematodes would be here. So I would cut this open, put it in water, and then those nematodes should emerge, and I could be able to identify them under the microscope. Usually you can identify them to genus and determine the number and population per weight or volume of soil or plant sample. The numbers is what's going to determine the severity of the problem, because there are nematodes everywhere. Okay, so we just have to look at the population to see what would be a damaging amount. So in order to control these, what's going to happen is, you can see on On page 79, under figure 4.16, they show you a sampling pattern, which they show two rectangles, and one is a sampling pattern that goes diagonally from one corner of the rectangle to the other, and then the second pattern kind of zigzags through the rectangle, so you can try to get an overall assessment of the total infection or the amount of nematodes on that site. And then they fill the on a turf green, they would fill the soil or the holes back up with the same soil that's on the green. You're never going to eliminate all plant pathogenic nematodes any more than we could get rid of all insects in the area. Depending on what the damage potential is, so you look at the number per 100 cubic centimeters of a soil sample, and actually for creeping bentgrass golf putting greens, they have a level listed on page 79 for how many of each one of these specific nematodes are damaging to golf greens. And this is actually creeping bentgrass. So basically, once you look at them, these are the damage thresholds they've established. And so basically, if you get something like that and they're above those thresholds, then perhaps it might warrant treating. Or you could change your maintenance practices or something that's allowing those nematodes to thrive and exist. And this would be a picture of a soybean cyst female. Again, I'll get them. These are the females here coming. They're bursting out of the stem. And this would be the nodule on a soybean root that it actually is nitrogen fixing bacteria in it. So there's the nodule, and then these are the little cyst nematode females that are bursting out of the stem. So they have eggs within them. Okay, sometimes there are problems with these in greenhouses. We can actually get rid of the plant debris. I already talked about pine lumber and wood chips going through a kiln to heat everything up. Steaming is a good way to get it out of soil. If you're making them in a flower bed, you can steam it. Some of the processes they've used too in flower beds is solarization. So you take a sheet of plastic and put it over the bed when you're going to have high sun, and basically it should heat the soil and reduce the nematode populations at a pretty shallow depth, but for most flowers that might work. In a lot of our, one of the things you can do is by cleaning pots and benches with either heat, steam, or sometimes they'll use a 2% bleach solution to wipe down the benches, so it would kill any nematode eggs or populations that might be there. And then potato, or I should say not potato, but in a lot of cases they use crop rotations. The reason I was thinking about that, the potato cyst nematode populations can actually decline 30 to 50% annually when a non-host is grown. And if they have 7-year rotations, you probably would never have economic damage from them. So there's a picture on page 81, which I did not get in my slides, but it shows a reduction in soybean cyst nematode damage to soybeans in areas left of the picture where they have corn and peanut non-hosts were rotated with the soybeans, and then on the area on the right where you had continuous soybeans, there's just severe damage. And one of the little factoids they have in here as well is that the tear gas or chlorpicrin is a soil fumigant that's used to manage nematodes in a number of different crops. And then the other thing, and we saw these on page 81, it talks about the beneficial nematodes, and we saw that in the nematode video clip that I showed yesterday, that Heterorhabditis and Steiner nema are pathogens of insects and are considered beneficial nematodes. And these are the ones that can be purchased for control of soil insect larvae that damage roots of crops. Okay, and I think I just have 2 more slides here. And this one is just showing, and I put the whole screen up here. It's not really, there, I'll bring that up. Applying methyl bromide to a field as a soil fumigant. And here they inject the methyl bromide under the plastic so it keeps it in. these were actually phased out in the United States back in 2005 because when it dissipates, it's a threat to the ozone layer. But there's some crops where they have to use this. I told the class in class that I had students in Florida, because we have such sandy soils, that they made a good living out of fumigating. That's all they did was fumigate for nematodes in Florida. But this was back in the 80s when it was still legal to do something like that, and it was probably the only thing we could use at that time. There are other fumigants. I had my fumigation license, and I actually used a fumigant where we would apply it as a granular to the soil, although I did wear a gas mask with that because you never know what fumes are coming off of it. We would till it in and irrigate to make sure that it was activated and irrigated again. And then as soil temperatures, we would till it again after it had set for, I'm trying to think, it was probably 2 weeks. We'd till it again and allow that soil to air out, but it was a minimum of 2 weeks. That you have to read the label and see what the minimum time is for each one. Okay, so you can use these fumigants on areas where you don't have plants growing to reduce the populations, but there are also some non-fumigant nematicides that can be applied to established plantings. And most of these are nerve poisons, toxic to nematodes, insects, and living organisms with the nervous system. So that means they're going to be toxic to you too. And then we have some other parasites and predators of nematodes, which can actually be a potential for biological control. And I've got a picture of one here that I took out of the text as well. And this is a fungus that actually traps nematodes by using their hyphae to create a noose around them and just strangles them. So you can see here, they've got one area where it's just the noose has gone around and it's just cut off the exit for anything coming through the nematode. All we need would be another one up here in his esophagus and it would be dead. So basically there are some, there's research going on to repel nematodes with plant extracts, cover crops, companion crops, green manures, which is the one article that I actually handed out to you on mustard cover crops, which I need to post in my other class, but they're already posted up online here. And then the other thing you need to think about is genetic resistance, because we do have crops that are bred, certain cultivars that are resistant to root knot nematodes. And on the left, you've got a lettuce that's resistant, and on the right, you've got a lettuce that is susceptible. Excuse me, to root knot nematodes. So this is actually, genetic resistance is one of the most effective for nematodes that required specialized feeding sites, so you can work on the roots of crops. So this takes a lot of monitoring and breeding to be able to get something like that. So your management choice is going to be based on soil and plant assays to determine whether your nematode populations actually justify the cost in some environmental impacts you could have with any management strategy that you might employ. So probably one of the main thing is we can delay the spread of nematodes by quarantines. Once we have nematodes present, we need to look at what management issues might help get rid of them, whether it's heat treatment and chemicals. But sometimes we have restricted use in particular areas. So one of the best things we can do is create optimal growing conditions that will help our plants tolerate the presence of nematodes. Because if they're not under stress, it's going to take a lot more nematodes to cause any problems. So some of the main things we can do is provide adequate water and fertilizer. Don't use pathogen-free propagation parts if we're using plant parts to start our crop. We can actually rotate to non-hosts, remove weed hosts, destroy our infected plant debris. All these are listed on page 83. I talked about solarization, steam the soil, fumigate the soil, applying nematicides, and again, plant-resistant hosts. So that's where I'll stop for today. Thanks for your attention.